So at UVA we have a biosafety committee, and they review all protocols that have to do with biosafety and recombinant DNA practices. And nationally, when I was looking up this information, there really isn't an oversight committee. So there are the NIH guidelines on recombinant DNA, and from what I understand, those may be needing revision as well, because I think with the advent of more genetically modified organisms, certainly in bacteria and other microbes, and you think about using gene therapy, for instance, viral vectors for gene therapy, which is sort of gaining acceptance that this would be a way to help people who have genetic problems. This is I think we need to rethink exactly what these guidelines meant. The recombinant DNA guidelines are from the 70s, and so it's when everybody was afraid of the way that bacteria would be getting out into the world and getting everybody sick or something. So, in general, I think there needs to be better education about and an outreach about what genetically modified organisms mean. And certainly people think about crops, they don't necessarily think about bacteria. And since the general sentiment is that bacteria are bad, people don't necessarily think about the fact that you can use, and engineer these bacteria for good practices like your team is trying to do. So, in general, I think there needs to be a different response in terms of oversight of this work, and then a way to educate the public better about what these practices actually mean and that these can be useful.
There's actually several different mechanisms by which control of recombinant DNA research is dealt with at the university. So there's a committee that oversees all of the activities that are done in laboratories, and this is out of environmental health and safety. And so you have to register every year the kinds of studies you're doing and the kinds of organisms that you're working with. And so if you were working with recombinant DNA, you have to declare that you're working with recombinant DNA. You have to declare whether or not you're working with only partial organism genomes or whole genomes from organisms. And then it's determined at what biosafety level you're at. So you're either biosafety level 1, biosafety level 2, and so on. And that deals with the kinds of containment that you have to have in order to protect not only the people that are working in your lab, but also then anyone else at the university. And so there's very clear mechanisms that the university has in place. In terms of ethical activities, if you're doing work that's associated in any way with humans or could have an impact on human health, and there's any human subjects involved, there's institutional review boards, IRB committees, both in the social sciences and in the general sciences, that review your protocols.
Well, of course there's a risk, and in spite of everybody's rock solid assurances that nothing could ever escape, there's always some kind of hypothetical risk, and sometimes it turns into real risk. There is an approach to risk that would basically take hardly any risk, the so-called precautionary principle. It goes under a lot of guises, and it comes in various flavors and strengths, right? So simply talking about the precautionary principle doesn't really tell you much, but one understanding of it is that before you venture, to muck around in this area, you have to satisfy everybody about the potential risks being attenuated or eliminated. Now, I personally think that's way too high.
I mean, so horizontal gene transfer would be the major one, because bacteria we know can readily exchange genetic information, and one of the concerns, of course, is that if you're engineering a bacterium and you're engineering into it as a way to make your modified strain some sort of selection, like with antibiotic resistance, that antibiotic resistance gene could escape from the bacterium and then get transferred to another bacterium. And actually yesterday, I don't know if you saw this, the Centers for Disease Control put out this huge press release about antibiotic-resistant bacteria as this major public health threat in the U.S., and they specifically listed different bacteria to be aware of, that these are problems. So I think when creating a genetically modified organism, that would be one concern to think about. The other is that once you release something into the world, you have no idea what's going to happen, and so that bacterium is alive and has some mutated gene. You can't predict how that would influence the activity of another bacterium that might be able to access that gene. Now, the risks are relatively low, but they're still there, and I think that given the backlash against, say, GMO crops, because that was a concern there, I think that people are, that would be something to consider for genetically modified bacteria. Of course, the issue becomes that we've managed to genetically modify a bacterium without any intention, right? So the overuse of antibiotics and misapplied use of antibiotics has allowed antibiotic resistance genes to spread. We haven't done it directly, but that pressure has allowed them to spread. So at least this way we can control what's being released. But I think, if you want to start talking about the mini-cells, that is the advantage of your mini-cell system, because there is no nucleic acid that would really be capable of escape into the world. So,
well, it's an imprecise science. It's always based upon the notion that you can predict the outcomes to maybe 99%, but that 1% of uncertainty that's going to occur is always a risk. Right? So you try to limit your risk by designing experiments that would have, I think, a reasonable chance of success. Right? No one is going to just take a whole portion of a genome and slap it into an organism and say, "Oh, let's see what happens." That's unlikely to either get approval or be the kind of science that you would expect legitimate scientists or scientists who have some semblance of a brain to carry out. Yet you never know what's going to happen when you do a transgenic organism, because even though you take what appears to be a relatively benign gene sequence from one organism and transfer it and expect it to function in exactly the same manner. So, for example, if you took a protein kinase, you would expect it to function as a protein kinase if you moved it from an E. coli to a mouse, or from a mouse to a bacteria, or however you're moving it through organisms. You don't know for certain whether that same molecule is going to have some secondary function that you weren't aware of. And so the potential always comes is that when you do a transgenic organism, you have to be able to, as I said, limit the risk by looking at what you think is going to happen, but also be aware of sort of off-target sites. And you see this occur, and people will report this in the literature, that they were looking at one set of outcomes, and they happened to notice that there was something unusual that happened also as a consequence.
Some of the more conservative versions of the precautionary principle are probably way too conservative, but there are real worries here. And synthetic biology has the potential to introduce entirely novel organisms into the environment that are self-reproducing. And it's difficult to say where that's going to lead. So it does call for, a really significant amount of caution. I think that iGEM is really quite a positive force. I really do. First of all, they require some ethics instruction or, ethics concentration. People have to think about the ethics of what they're doing. What I also like about it, is this BioBricks thing, where they basically pass these out,
Right.
To any sort of team willing to participate according to the rules. And one of the rules is that, you have to make whatever you produce available to anybody who is interested, right?
Mm-hmm.
I think it's the good, really it's kind of the reassuring, altruistic good side of synthetic biology. On the other hand, not to cast aspersions at anybody, but you've got a highly entrepreneurial.
Well, so the fundamental logic behind iGEM is great, right? So you have the notion that people who create modules or cassettes or genes that are of general value should openly share them, and then be able to use them in an open format in order to create new ideas and new products and new innovations. And so from that standpoint, iGEM is great.
There is always this pressure between what scientists and researchers are discovering in the lab and their willingness to share, because in general people do want to share this information, as compared to the need for proprietary information, for patenting it, for making it owned by a company or a university or whatever that is. And so I think in terms of rules and regulations, that might actually be something that needs to be revisited in many ways. I think the patent and copyright offices and trademark offices at universities don't necessarily think about this. It's much easier to think about patenting a machine or a process than do you patent a BioBrick, do you patent a Minicell? And that's completely, I guess, antithetical to what iGEM's trying to accomplish. But you did create something, and so what is the proper recognition of that? Is it just that, you guys, if you were to create something as a BioBrick, somebody else uses it, you get an authorship, you get cited,
As I understand it, that Supreme Court case said you couldn't patent genes that occur in nature.
Right.
But if you tweak them in your lab in some way,
Right.
then they're patentable. So I would assume, by definition, a lot of the stuff that synthetic biology will be coming up with will be de novo, it'll be new stuff, really new, not found in nature, and that's the whole point of it, right? To control nature through a combo of engineering and biology. So that, there's a big space open there for entrepreneurial activity.
I think from the standpoint of good practices, right, it teaches people responsibility, right? And I think that's the one thing that's really important, that by sharing and not limiting access, you understand that anything you do should be available for other people. Whereas if you think about the way people have looked at patents and patent protections, it's meant to benefit the people who have put the investment into it. Right? That's the whole idea behind a patent, is you want to recoup something that your research has generated. On the other hand, if the end point is to help society broadly, right, you have to be cognizant that there has to be flexibility within those limits. And so we work a lot in the developing world in my laboratory, and there are stipulations for all of the work that we do, in which you may seek patent protection within the context of a developed nation, but you can't limit the use of your discovery in the developing world. And I think a lot of corporations are adhering to that. That is, if they're competing, if you're Monsanto and you're competing against Brazil and Argentina, you want to be protected in Brazil and Argentina. On the other hand, if you want to have an impact in sub-Saharan Africa or in parts of Asia, then you want to not restrict the use of those discoveries. And so I'm not certain where iGEM fits in terms of patenting materials that you come up with, but my assumption is that everything that is done through the iGEM mechanism is supposed to be freely available worldwide, which is really noble.
So what do you think about then tailoring these kinds of therapies to an individual's needs, like in gene therapy?
Well, I think that's a really tantalizing question,
Yeah.
really interesting, okay? Because I think, it's becoming pretty clear that, the standard approach to cancer therapy, for example, is this big, one-size-fits-all, and that hasn't worked out terribly well, right? And so people, researchers and clinicians are getting much more sensitive about getting some kind of genetic match, between the individual, so personalized medicine, get an individualized match between the patient and the chemical compound that you're giving them. And that's all to the good. Although I kind of wonder about talking about entrepreneurial issues, I wonder about the cost effectiveness of that, right? Because and this is way beyond my pay grade. This is way outside my area of expertise, but I'm assuming that drug companies make a boatload of money, by producing drugs that will be, administered to, hundreds of thousands of people, right? That's where the money is. So I just don't know what's going to happen with personalized medicine. It's a really interesting project to sort of mold a particular treatment to a particular patient. It sounds really ideal. Whether it's a money-making, given the small scale of it,
Right.
Given the money, the hurdles in terms of profit-making off of. I just don't know what the economic model, the business model will look like, okay? Because if it really is just very close tailoring of the individual, the individual's genetics to the drug you're giving them, that could have problems of scale, It's like each one's a one-off,
Yeah.
and that might not be a really attractive business model.
Mm-hmm.
We had talked a lot about what the application of a Minicell would be, and you all felt like it would be something that could be applied to almost any circumstance, a drug delivery circumstance, a vaccine-based circumstance, because of just because of the technology behind it.
What kind of impact it's going to have on the body, it might not replicate, but it could, conceivably could mess people up.
Yeah.
But I have no real expertise. The only point I would make is that it seems to be a really nice method for doing things.
Mm-hmm.
I see that as a real future avenue for medical progress.
Okay.
Non-replicating is a definite benefit in terms of safety, but there might be other ways in which it's conflicts with human health.
Sure.
That's for us to find out.
I think that the Minicell concept is great because it avoids the problems with recombinant DNA and genetically modified bacteria that we were talking about earlier, because now you have this Minicell that's budded off from the bacteria that does not contain any nucleic acid that should be capable of exchange to another microbe. But you have a Minicell that basically is a piece of its parent. And so it will have on its surface the kinds of, and carry within it the kinds of content that you engineer it for. And so we talked a lot about how to make these safer. So you're making the Minicells out of E. coli, and E. coli has some of the most toxic lipopolysaccharide, which is an outer membrane surface exposed structure that's important for bacterial bacterial overall sort of structure. And it has that lipopolysaccharide is recognized by our immune system, and it's known that even for the kinds of lab strain, non-pathogenic, not causing disease E. coli that you work with, these are capable of causing a major immune response in people that would lead to shock and potentially death. So how do you engineer a mini cell that still can carry that content and take advantage of E. coli's natural ability to create mini cells, and the way that you engineer it in a way that would allow that would reduce that threat to human health. And so we talked about there were a few ways that you approach this. One was to use a bacterial E. coli background that is mutated in its lipopolysaccharide structural enzymes, so the enzymes that build the lipopolysaccharide. So you're removing the parts that are toxic, and you were able to get hold of those from that strain from someone else. Is that right?
Yes.
So I think that's a major step in the right direction. The other was, how do you maintain that this mini cell is going to stay intact inside the human body? It's not just going to get, basically degraded by either by our immune cells or by proteases in the bloodstream, that sort of thing. And so the way we talked about that was to coat the bacteria with a protein that allows it to now be masked in a carbohydrate that is something that looks like something that human cells make. So the immune system has learned to not react against it. And so that is a very ambitious approach. And so if you can make that work, I think that is a really elegant way to avoid the problem, because it's something that actually the bacteria that can do that, it helps them have an advantage. And it's known from all kinds of studies that we can do in the laboratory that is a way that they can avoid getting engulfed and killed by cells of our immune system, and also degraded by these factors in our bloodstream that normally recognize these foreign bodies. So that was another approach. And then the third approach was the preventing complement deposition, right? So which the sialic acid that we're talking about would also do that. But then the other was putting a protein on the surface of the bacterium that would prevent these soluble factors in the bloodstream from attacking and basically blowing up your mini cell, for lack of a better term. And so, and the way we talked about various bacteria, and you're actually taking advantage of, this is something that a pathogenic bacterium uses to survive in its host animals, including people. But you can use this for good. So you can take this protein, which normally the reason it aids the bacteria to be pathogenic is not because that makes it sick. It just helps it survive in the body. And so you're taking advantage of that and putting this in this non-toxic mini cell, and so helping that mini cell to retain activity. And so, I think those three approaches all together are just fantastic.